Hey guys, and welcome back to Now I Know. Today we are talking about antigen processing and presentation. Now, if you remember in one of the previous videos, we have talked about humoral immunity and cell-mediated immunity. In cell-mediated immunity, we talked about T cells and how T cells on their own cannot recognize the antigen. What that means is they need to be presented the antigen by some other cell. So if a cell finds an antigen in the body, it needs to degrade this antigen. Probably this is protein that we are talking about, so it needs to have a small fragment of this protein, a small peptide, and it will present it to T cell. Okay, I'm just talking very crudely. I'm not even talking about what kind of MHC or what kind of a T cell it is, just very crudely. Whichever T cell it is, it doesn't matter TC or T helper cell, it needs to be presented with the antigen. It needs to be presented with the peptide. Then only it can recognize this complex MHC attached with a peptide or a portion of antigen, and it will mediate or it will carry out the further destruction of the antigen. If it is TC, there will be cytotoxic killing, or if it is T helper cell, there will be further cytokines production and so on. So no matter what kind of a T cell we are talking about, I always think in my head, always I feel they are little high maintenance. They are very efficient, of course, but they're little high maintenance. They cannot on their own go and carry out the killing or carry out the elimination of antigen. They need to be presented. So this whole scenario where a T cell is presented the antigen is what antigen processing and presentation. And in our body, there is a very specific way. There are two pathways that are present by which antigens are presented to T cells. Okay, now before I go further in detail, a little more introduction about this. So when we talk about T cells, as I said, they need to be presented the antigen by MHC molecule. Now we have talked about MHC class 1 and 2 molecule, the difference between both, and TC and T helper cells. I'll just give you an overview over here, just very briefly, 2 lines. If you want the detail about the whole thing, you can have a look at the video on our website nowiknow.co.in. So in short, what happens is, so if it is a T cell with CD8 on its surface, that can recognize only MHC class 1. And where this MHC class 1 is present? MHC class 1 is present on all the nucleated cells. Okay, we gotta remember this, all the nucleated cells. That means almost all the cells in the body will have MHC class 1. Which is the cell that will not have it? The cell without the nucleus, RBC. So other than that, all the nucleated cells will express MHC class 1. So TC cells, that means the T cells with CD8 can recognize the MHC class 1 that is present on all the nucleated cells in the body. When it comes to T cells with CD4, those are T helper cells. They can recognize only MHC class 2. And which cells express MHC class 2? Only antigen presenting cells. And which kind of cells are antigen presenting cells? Cells such as macrophages, dendritic cells, cells such as B cells, they are the antigen presenting cells, and they will express MHC class 2. Would they express MHC class 1, this antigen presenting cells? Think about it. Yes, of course. Why? Because they are also nucleated cells. So these cells, antigen presenting cells, will have MHC class 1 as well as MHC class 2, right? Because they are nucleated cells. But not all nucleated cells can express MHC class 2. Think about it, what I just said. So now there's a practical thing that you need to understand is, of course, how will you remember this? I used to get confused when I was a student, like how will I remember CD4 as what T helper cell and can recognize MHC class 2. So this is how I used to remember it. This is just a trick to remember. T helper cell, here if I write H, it has 2 lines. And if I write 4, I write 4 like this just for this purpose. So I have 2 lines over here. And class 2 of course, it has 2 lines. So helper cells express CD4, can recognize MHC class 2. Simple. And the rest is TC cell 8 and 1, whatever combination is left. This is about briefly about T cells and MHC molecule that we need to know. Just let's pin this aside. Next, we talk about antigen. What kind of antigens can this T cells be presented with? Now, when it comes to antigen, we will have broadly two classes of antigens, exogenous antigens and endogenous antigens. The term itself says exo means it is produced outside the host cell and it is taken up by maybe phagocytosis or endocytosis. Let's say, for example, there was an antigen or a bacterium that was present in the environment and macrophage has taken up this cell, and now it will digest and it will produce the peptide. Since it is antigen presenting cell macrophage, it will express MHC class 2 and it will present it to, since it is MHC class 2 antigen presenting cell, it will present it to T helper cell. When it comes to endogenous antigen, endo, the term itself says, that means the antigen is synthesized within the host cell. Now, why would a cell synthesize an antigen? That's something that we don't want. Why does a cell do it? So there are scenarios like, let's say for example, in case of a viral infected or virus infected cells, what happens in that case? Virus is infected and you know that virus can take over the host cell machinery and it will start synthesizing the viral proteins. So in this case, when the viral proteins are synthesized by the host cell, that is an example of endogenous antigen. Or there also could be a situation where the proteins are produced in the cells. You know, the proteins have half-lives. So it needs to be degraded at certain points. Also, some proteins, when they are synthesized in normal condition in our body, could be defective. It could be abnormal. It could be there could be a folding problem. So there could be a misfold or some kind of problem in our normal protein, regular protein, that needs to be degraded because they are abnormal or they have some fault in them. So in such case also it is called endogenous antigen that needs to be degraded. Also certain proteins, unique proteins on the cell surface. In case of cancerous cell, this is the proteins that it will express will be very different than our normal cells. So that is again an example of endogenous antigen. So these are the cells. They could be any cell that are infected with virus or any cell that could have problem with the misfolded protein or defective protein or abnormal protein, any cancerous cell in the body. So these are the cells that can express, because they all at the end are going to be nucleated cell, except for RBC, we are not talking about it. So all the nucleated cell, any of the nucleated cell, if it encounters any of this problem, because it can express MHC class 1, it is going to be recognized by TC cell. So as I said, the antigen could be a large protein molecule. The whole thing cannot be processed by the cells. So generally this antigen, this protein will be degraded into small peptides, and this peptides, this produce like the production or the degradation from protein to peptide, it's a whole event, and that is what is called the antigen processing, because we are processing the antigen to be presented into small peptides. So there has to be some kind of degradation. This whole thing is called antigen processing. We need to process the antigen, simple enough. And once this peptide is made, what are we going to do with that? We need to present it to T cells, and that happens with MHC molecule. So peptides that are produced will form complex with MHC molecule, will be transported on the cell membrane because it needs to present it to T cell. So that presentation to T cell is called antigen presentation. That is why it is antigen processing and presentation. This whole thing of antigen degradation and presenting it to T cell requires antigen processing and presentation. Since we have two different types of antigen, two different types of T cells, we will talk about two different types of pathways. So this is just the introduction of this antigen processing and presentation. Now we'll see the pathways that are involved in this processing and presentation. So in this video we are talking about the exogenous antigen or the exogenous pathway. The endogenous antigen has been already dealt in another video, so if you guys want, you can have a look over there. So when the exogenous antigen is being processed, the pathway is of course called the exogenous pathway or the endocytic pathway. Now, we just saw that when it comes to exogenous antigen, antigen is taken up from outside the cell. All right. And when it comes to exogenous antigen, antigen presenting cells are the one which will take up the antigen from outside and present it on MHC class 2. Now, this taking up of antigen could be either by phagocytosis or by endocytosis, or in some case some cells can perform both. Let's say for example, when it comes to macrophage, they can take up an antigen by both phagocytosis as well as endocytosis. But in case of B cells, they can carry out the receptor mediated endocytosis. That means endocytosis with the help of receptor. Let's say for example, look at over here, they will have antibodies on their cell membrane which acts as specific antigen specific membrane bound antibody that will act as a receptor. So the antigen will bind to this antibody and it will be internalized, and this is what is called receptor mediated endocytosis. So in whichever way the antigen has been taken up, what has to happen is it needs to degrade into peptides, right? Because fragment of that peptide will be expressed or will be displayed on the cell for T cell. And here we are talking about T helper cell because we are talking about MHC class 2. Now here I'm just briefly going to tell you how this degradation happens. The process of phagocytosis or pinocytosis we have talked in a different video, so you can have a look over there. So what will happen is, in case of endocytic pathway, once the antigen is taken up, the antigen is passing through the compartments in the endocytic pathway for the degradation. That is from the early endosome to late endosome to lysosome. And as it goes on, the acidic condition in the compartment increases. You see the pH is decreasing. That means the acidic condition increases. And this acidic condition is what causes the degradation of the antigen into peptides. And here the peptides that are formed are 13 to 18 residues. Now this number is important. Okay, I will just come to it why it is important. So it will produce peptides of 13 to 18 residues. Now as the antigen passes through these compartments in the lysosome, it is not only the acidic condition but also certain hydrolytic enzymes. About more than 40 or acid dependent hydrolysis are there that causes the degradation of antigen into peptides. So now once the peptides are made, that is when they are ready to be displayed on MHC class 2. Alright, so before we move on, one very important thing that we need to understand over here. We are talking about exogenous antigen. So we are talking about antigen presenting cell. We are talking about MHC class 2. So we are talking about antigen presenting cell. In introduction, we just saw that even if it is antigen presenting cell, even though it has MHC class 2, they will also express MHC class 1, isn't it? This is I'm showing on the surface, but I'm talking about in their endoplasmic reticulum. There will be assembly of both MHC class 1 and 2 is possible. They can express both, right? So if it is infected by endogenous antigen, of course it will take up the route of MHC class 1, go ahead and process it. But if it is taking up the exogenous antigen, it has to produce it on, or it has to express it through MHC class 2. So this particular cell will have both 1 and 2. How would it decide that this antigen is endogenous, so I should opt for MHC class 1 expression, or this antigen is exogenous and I should opt for 2 presentation? How would it know? There has to be some mechanism that helps the cell to either take 1 or 2 based on the antigen that is produced. That is where this peptide size that is produced comes into picture. If you have seen the other video where we talk about the endogenous antigen, the peptides that are produced are 8 to 10 residue long, and the exogenous antigen peptides are 13 to 18. So there is quite a big difference. Now how this helps? So what happens in the endoplasmic reticulum, the MHC class II, the binding cleft where the antigen or the peptide binds, that cleft is always physically blocked or occupied by something known as invariant chain. In short, the MHC class II, the cleft where the peptide has to bind, is always physically occupied or blocked. MHC class 1 is open but 2 is occupied. You know that MHC class 2 is available only when there is exogenous antigen. But so if there is an endogenous antigen, it has only MHC class 1 where it will go and bind. But there is MHC class 2, it is blocked. But in case of exogenous antigen, the same thing could happen. It can go and bind to MHC class 1. So what happens is, that is where this size of peptide is very important. The cleft of MHC class 1, since it is only specialized or it is meant for endogenous antigen, the cleft is smaller. Okay, it can occupy or it can take up peptide only of maximum 10 residue bigger. You know, it's only meant for something like that. Whereas in case of exogenous, the peptide starts, the minimum itself is 13 residue. So in short, the peptide is larger than the endogenous that cannot physically sit in the MHC class 1. So of course the exogenous antigen is not going to sit on MHC class 1. So only endogenous will go ahead and bind. Once it is bound, it will go ahead and carry out the presentation to our TC cell. But when it comes to MHC class 2, since it is made for bigger residue, of course the smaller can go and bind, right? The endogenous also can bind over here. So in order to avoid the binding of endogenous to MHC class 2 by mistake, there has to be some mechanism that physically blocks the MHC class 2 cleft for peptide binding. That is why it is always blocked by invariant chain. And that can be degraded only when there is exogenous antigen, and we'll see how it works. So you will see that in endocytic pathway or in exogenous antigen pathway, we have this invariant chain that we talk about, and you should know how and why this exogenous and endogenous, how a cell differentiates, because in case of antigen presenting cell, they can express both. Now let's move on. I think now when we talk about the exogenous pathway, it will make sense. So you see, when it comes to MHC class 2, it will be physically, you know, the site where the peptide bind is physically always occupied by invariant chain. It makes sure that the cleft of MHC class 2 is always occupied, and only when the exogenous antigen is taken up, it will be degraded. Now we'll come to see what is this clip and all. So as I said, antigen presenting cell can express both 1 and 2. So there has to be some mechanism that prevents class 2 from binding with endogenous antigen. And because of the residue, I told you the size of peptide is small in case of endogenous. So it can easily come and sit on the MHC class 2 cleft. So in order to avoid this situation, we have something called invariant chain. That is nothing but a trimeric protein that will interact with the peptide binding cleft of MHC class 2. And thus it will prevent the binding of any endogenously derived peptides. And it's now easy also to understand why this APC doesn't get confused whether to go for MHC class 1 and 2. Now, as I said, the proteolytic activity or the acidic activity in each compartment, starting from the early endosome to the late endosome to lysosome, increases. So gradually, as the antigen as well as the MHC class 2 that is passing through this mechanism will start undergoing certain changes. The antigen will be degraded into peptide, and MHC class 2 has to undergo this so that this invariant chain also will start getting degraded. So this is what is happening when the antigen is, let's say for example, this is the exogenous antigen that has been taken up and it will be degraded. Okay. Now this degraded antigen will combine with the MHC class 2 that is separately being routed towards this degraded antigen compartment. So this MHC class 2, when it is getting transported through the endosomal compartment, the same process will happen over here. The pH will reduce and it will break down the invariant, or it will degrade the or digest the invariant chain. Separately here you can see the antigen has been also taken up going through the endocytic compartments. Here only one diagram is shown, but there will be a process of early endosome, late endosome and lysosome, and because the pH is decreasing in this endocytic compartment, it will digest the invariant chain. But this is not completely digested. There will be a small clip. This clip stands for, here the clip stands for class 2 associated invariant chain peptide. Okay, a small fragment of this clip will be remaining, and to remove this clip there has to be a presence of exogenous antigen. Only when the exogenous antigen is present, it can physically remove this short clip. In this particular replacement of clip by the peptide, we need another molecule that is called HLA-DM, human leukocyte antigen DM. It is non-classical MHC2 molecule. We require HLA-DM, and this is going to facilitate the exchange of clip for another peptide. Now what has been observed experimentally, that if there is another molecule called, or the another non-classical MHC molecule called HLA-DO, if this is present, it is going to negatively regulate the mechanism of HLA-DM, and in that case what will happen, that clip will not be replaced. So the peptide cannot sit over here. So HLA-DO, you might read this, can negatively influence the mechanism of HLA-DM. And if HLA-DM is not able to, or if it is not present, cannot facilitate, or it cannot help the peptide to replace the clip. So in short, binding of the peptide to MHC class 2 involves a replacement of invariant chain, or a part of that invariant chain that is left behind, that is clipped, class 2 associated invariant chain peptide. And that particular mechanism, that replacement, is catalyzed by the help of HLA-DM. And you can remember one more point, that HLA-DM is negatively regulated by HLA-DO. So HLA-DO acts as a negative regulator of MHC class 2 antigen processing, and it binds to HLA-DM, and thus it will inhibit its role in dissociation of clip from MHC class 2 molecules. So it's been observed, or it's been seen, that why this HLA-DO could be having this effect is because it might have a role in selection of peptide that binds to MHC class 2 in B cell. Only an appropriate peptide is present, it will let HLA-DM to replace the peptide for clip, otherwise it will inhibit the mechanism. So this is what happens in case of MHC class 2. An exogenous antigen that has been taken up will be degraded into peptides of 13 to 18 residue by going through the endocytic compartments where the pH is decreasing, and that will cause the antigen to form peptides. Simultaneously, we'll have MHC class 2 that is physically bound with invariant chain going through the endocytic pathway or endocytic compartments, and that will result in digestion of this invariant chain. Now you know why this is present, and leaving behind the clip. And this clip is present because only when an appropriate or mature exogenous antigen peptide is present, it can replace the clip. And for this replacement, for this reaction or the exchange to happen, there has to be a presence of HLA-DM that facilitates this replacement. And there is also one more molecule called HLA-DO that negatively regulates the HLA-DM mechanism. So what happens as a result? This MHC class 2 is now expressing part of exogenous antigen on the membrane of antigen presenting cell, which will be recognized by T helper cell. That's all how an exogenous antigen is presented by the antigen presenting cell, and we also understood how it does not get confused whether it is exogenous or endogenous antigen. So that's all for now. I hope this video was helpful, and I will see you next time. Until then, keep learning.